Great. Welcome back to our second session. And it will be chaired by Steve Kregel. Steve is Associate Director of ISI, and he's also Research Associate Professor of the ECE Department of Veterinary School. Okay, Steve.
Thank you, Joshua and Yolanda. So welcome to the next session, which is on neuromorphic computing. We have 4 papers or 4 talks today, 3 of which have to do with the integration of biological systems and electronic systems. And the 4th one is on a biologically inspired electrical system. Our speakers today are Maryam Shanechi from the Electrical and Computer Engineering Department, where she's Associate Professor and has been with us, I think, since 2015, if I remember right, also in the biomedical engineering and computer science departments. Yasser Khan, who joined the Electrical and Computer Engineering Department as an assistant professor last year, where he came from Berkeley and Stanford. Dong Sung, who's a research associate professor in biomedical engineering and has been with USC, I think, since about 2005, if I'm not mistaken. And finally, Akhilesh Jaiswal, who is a research assistant professor in the ECE and research scientist at ISI, where we're glad to have him. And he's been with us, I think, since about 2020. I apologize for doing that from memory, but I think I'm close. So with those introductions done, Maryam, I'll turn it over to you for the first talk.
So hi, everyone. It's a pleasure to be here today and talk about some of the work that we do at the interface of AI and neuroscience to develop next generation neurotechnologies. So as you may know, brain disorders are a leading cause of disability, and unfortunately, we are pretty bad at treating them. For example, up to about 30% of people with major depression do not respond to any of our current therapies, whether medication or psychotherapy. So based on the understanding that these disorders are signs of abnormal electrical activity across large-scale brain networks, an idea that arises is whether we can revolutionize therapies by trying to regulate or control these abnormal network activity patterns. And there's indeed been proof of concept demonstration showing that if we electrically stimulate an appropriate region of the brain, depression symptoms may be alleviated in some patients. But the problem has been inter- and inter-individual variability, and actually the clinical trials of these disorders have shown inconsistent results across different individuals, which raises the question of how can we improve efficacy. Now, when we started thinking about this problem as engineers, our first intuition was that we have to develop systems that can address heterogeneity, inner and intra-individual variability in symptoms, in neural representation, or in response to therapy. So what if, instead of having a stimulation device that just turns on and stays on, we guide how we stimulate the brain by the very neural activity patterns that we are trying to control, and by a dynamic model of how the brain responds to stimulation therapy. Now, as engineers, we're pretty good at building these closed-loop control systems, for example, autopilots. So you may ask me, so what's the big deal? Why can't we do this for the brain? And unfortunately, in the case of the brain, of course, we didn't design it. It's an immensely complex system of over 80 billion neurons with unknown dynamics. And that's where we come to the question of machine learning to build data-driven models of this complex system and to come up with data collection frameworks that can enable this modeling. So what do these data sets or signals that we record from the brain look like? I'm showing you electrodes that we implant on the surface of the brain. These are called ECOG, and these are the signals that we record from them. You can see that there are many of these signals, giving them very high dimensionality. They're complex, they're noisy. There's no clear pattern in these data sets that would, for example, tell me how somebody's mood is changing in depression. So modeling, decoding, and control of these complex signal patterns is what is required to develop the next generation of neurotechnologies for regulation of abnormal states to treat a diverse set of disorders. And that is exactly what our lab focuses on. We work at the interface of machine learning, stochastic control, and neuroscience to enable decoding and control systems for neural activity and develop these next generation brain-machine interfaces. Okay, so what do these interfaces that we envision look like? We envision a system that would take these neural data sets, decode from them in real time an underlying symptom, let's say depression symptoms, and then use that as feedback within a controller that would then adjust the stimulation parameters such as amplitude and frequency of an electrical stimulation. Now, this model-based system that I've depicted here has not been developed to date, and there are multiple computational and machine learning challenges that we have to overcome to design the system. The first challenge is to develop encoding models that would tell us how a given state, let's say mood, is even represented in these neural data sets. But that's not enough, because even if I can encode and decode perfectly, I still don't know how to modulate. So I additionally need input-output models that would tell me the response of the brain to these input patterns and therapies. And finally, I have to combine these models to come up with a control strategy. So I'm going to show you very briefly some of the progress we've made in recent years, just to give you a flavor of how AI actually comes in and makes a fundamental difference to solving of these problems. So first, a couple years back, we developed machine learning models that enable dimensionality reduction to allow us for the first time to decode human emotions and mood variations from intracranial brain activity, thus opening the door to obtaining personalized feedback signals to better provide therapies. We also worked on the input-output modeling framework, showing in a recent paper that we can indeed learn models using machine learning that can predict the response of multi-regional brain networks to stimulation. We also developed novel stimulation patterns that were stochastically changing and exciting the brain activity so that we could come up with informative data collection frameworks. We've been addressing the challenge of multimodal modeling by recording activity across multiple spatial temporal scale in the brain and showing in some of our recent work that by aggregating and fusing information across multiple modalities in our model, we can do better decoding. We've been looking at shared representations and disentangling representations in neural activity that may exist of multiple behaviors, let's say emotions and movements. We want to disentangle those representations to better decode the specific behaviors. And we've done that both with analytical models as well as recurrent neural network models that can do the disentanglement. We've also been looking at nonlinear representations, and in particular, based on the biological insight that these representations in neural activity may actually be evolving on nonlinear low-dimensional manifolds that involve loops and holes. So we've been developing geometric tools to learn this topology and geometry and to explicitly incorporate it within dynamical models. So these are just some of the flavors of what we do. In everything that we do, we always keep in mind that at the end of the day, we want these to have impact. We have to be able to implement them within hardware devices. So all of our models are amenable to real-time implementation and control. And an objective that we have is that eventually we will incorporate them within current and future hardware devices. So with that, thank you for the invitation. I hope I gave you a flavor of what we do with AI, and I'll thank my students and the funding sources.
Thank you, Maryam. That was right on time and very good, very important application and good work. I think the format is that we're going to save the questions for the end of the session. Yolanda will tell me if I'm wrong. I see one question from Priyanka in the chat, so we'll get to that at the end. And please, everybody else, keep questions coming in the chat, and we'll get to those. Otherwise, next up, Yasser's going to talk to us, and we're going to move from the brain to the skin and electronic skin. So I'm interested to hear more about that, as I'm sure all of us are.
Can you hear me?
Yep, sounds great.
Okay, perfect. Hello, everyone. So I will talk about more sensors, and essentially these are the new types of sensors. These feel and also act as a second skin. Essentially, the drive here is to make electronics, they're similar to the skin, and then they will give you new types of sensing capability that exist. And also the new sensing can be, I'll talk about the different sensing modalities. So we talked about the edge devices, and one of the dominating edge device is now wearables. And typically, if you think about wearables, you think about Fitbits and Apple Watches, but you can actually have much more functionalities in new wearables. So what I typically do is you can think about wearable sensing in essentially 3 different domains. One is bioelectronic, where you're dealing with electrical signals. Another is biochemical, where we try to get the chemical markers. And then finally is with optics, biophotonics. In the electronic side, you have same as what Maryam was saying, ECOG. If you are doing non-invasively, you can have EEG. There are sensors that looks at heart activity, that's ECG, muscle activity, EMG. And then the new sensing is becoming popular. It's biochemical, where we are looking at analytes in different bodily fluids, such as sweat, saliva, tear. So here we deal with lots of metabolites, hormones. And finally, for photonics, this is a non-invasive sensor that looks at oxygenation of the person using light and a light detector. And interestingly, you can actually put all of these things in soft and flexible devices, and they go around the body and collect the biosignals. What we envision will happen in the future is we will see a change in the hospitals. Right now, if you walk into the hospital, you'll see large machines. They're collecting all the biosignals. And in the future, we hope to go to a point where we will be using more wearables, implantables, and ingestibles. And they typically have the same functionality. Say ECG, I mentioned, you can actually use a wearable for that. Oxygen saturation, temperature, heart rate, respiration rate. Most of these signals we can actually get from flexible and stretchable devices, and those goes on the body, and we can break out from this form factor where things are mounted on the wall, large scale, to much smaller scales. And what are the benefits you get from these flexible devices? I typically give this example. So in one of our projects, we were trying to look at the skin impedance to implement a smart bandage. You can think about having a bandage that looks at the electrical activity of the skin. And whenever there's a damage, there is a change. But if you use rigid PCBs, they don't conform well to the skin. As a result, the signal to noise ratio goes down. But if you have a device that is plastic that folds around the skin and then gives you a good interface, we can get a good signal to noise ratio. Same goes for optical sensing. As I was mentioning, like if you have a device, an optical device that wraps around the finger, where it essentially reduces the ambient light, you will get a better signal. And we are now coming to form factors similar to tattoos, where the sensors can be printed on tattoo paper, and they have the same feel of a tattoo paper. even inside the body, we can put things and they can have a degradable aspect to it. So it degrades over time. So these are all wonderful. And what we are doing here at USC is basically we are looking at printing. People typically are familiar with photolithography for making the electronics, but you can actually print devices, and that gives you much more flexibility and versatility. We can inkjet print, we can blade coat. It's similar to spreading butter on top of bread, but the inks are functional and you can make functional optical devices. Screen printing, you can use that for making electronics, similar to how we print t-shirts. So with printing, we can actually go from device scale to actually like energy sources. So typically, we are 2 orders of magnitude apart from the state-of-art lithography. So we can think about in the micron range, the transistors and the sensors. So aligning with the topic today, I wanted to give one example where we have an exciting application and we are making the device, and then that device is coupled with state-of-the-art algorithms to do classification of signal in real time. So this is how we make an array of sensor that goes on top of the skin, especially on the arm. So you can think about printing the device on a printer. So here we have nanoparticle ink getting printed, and then we are making an array. We can make large arrays with printing. So you'll see like how big of a scale of devices we can make. And good thing here is since you're printing, you can do large scale devices. And then we can couple that with electronics that can run tiny ML algorithms. So here we use the hyperdimensional computing to train a model where it looks from the muscle signal what gestures you're making. And once you have the model with the gestures, and then it can do real-time classification. So that's wonderful because here we are essentially using new technologies to make the devices that didn't exist. And then we are coupling that with algorithms that exist, and we can do real-time classification. And it will be enormously powerful in the future where we can think about new sensors. So this is another sensor. It's I was mentioning the biochemical sensing. So this is a sweat sensor where we have transistors functionalized to look at analytes in sweat. And we can make this structure where we can look at different analytes, let's say sodium, ammonium, calcium, how those are changing. Since I don't have time, I'll quickly go to the demo part. Typically, the sensors we make are similar to a plastic. This is a tri-layer plastic. And here you can see like it has the openings where the transistors are. And these are functional transistors that looks at different analytes. And then we couple that with a existing embedded system. And then interesting part is the size of the battery. When we are getting so much data, but we are limited by the size of the battery that's in a smartwatch, we have to think about how much data we want to send to the cloud. Here I'll show basically like this one is a 140 milliamp hour battery, similar to what you'll see in Fitbits. And we can run and collect all the sensor data for 4 hours with such battery. But if you have ML running and then like the feature extraction is done in the sensor itself, then we can actually think about a huge battery boost where we can run this device almost 24/7. Typically, they go on the skin. So here is what I'm wearing the device on my forehead, and it's essentially collecting the optical data. And then we have the various sweat sensors that are also getting the sweat signals. This is also a fun experiment where you can see like the fluid flows into the channel, and then we can actually look at the response of the rate sensor, and it is increasing. All right, so to wrap up quickly, what we need right now is basically, if you look at the enormous amount of raw data we have in such new kind of wearables, we need to implement ML within the device, or there's a possibility it's like to look at new types of skin-like electronics to do the classification. So here I'm just showing that the wearables will get more complicated over time because we are looking at multiple modalities. And if you want to get data from the neural signal, the optical signal, the electrochemical signal, the physiological signal, it adds up. And it adds up in a way that you need much more computational power that a small microcontroller can handle. One of the possibilities we are exploring right now is we could think about having a similar transistor architecture and then doing the classification near the sensor with these organic electrochemical transistor or organic transistors, but the models need to be down to a level where we can actually deploy such models. I'm running out of time. Okay, so I'll wrap up. So basically, we work at the sensor and systems levels, but we are really interested on how to look at the data and process the data near the sensor so we could use it in real applications. Thank you so much.
Thank you for staying on time and keeping us on track, and for the great talk. We still have one question in the Q&A session, so please do enter your questions in real time so that we can come back to them at the end of the session, but please do get them in while your questions are fresh in your mind from these talks. Next up, we have Dong Song. We're going to go back to the brain. We went from the brain to the skin and now back to the brain and the hippocampus. So Dong, please go ahead and take
Okay.
away.
So today I'll talk about our effort on developing a hippocampal memory prosthesis. So here's the vision. The prosthesis is a biomimetic device that mimics the signal processing function that's normally performed by the hippocampus. So here, this animation shows like in the normal hippocampus, it transforms short-term memory information into appropriate information to form long-term memory. But once hippocampus is damaged, short memory function is still intact, but the hippocampus cannot generate new long-term memories. And our vision is to build this biomimetic device that records from the upstream region of hippocampus, does the appropriate transformation, and generates optimal stimulation patterns and stimulates the downstream region of hippocampus. And by doing that, we restore the signal transmission property and hopefully also restore the cognitive function. And in this case, the episodic memory function performed by the hippocampus. So this approach holds the promise of helping patients with brain damage, for example, like Alzheimer's disease patients or traumatic brain injury patients. So there are two important components of this approach. First, we need to have an interface, which I won't cover today. And the second is that in order to describe the proper input-output transformation, we need to have a computational model. So this approach is different from sensory prosthesis or motor prosthesis, which has observable input or output patterns. Since hippocampal is a very deep brain structure, both the input and output signal of hippocampus are spatial temporal patterns of spikes. So the modeling goal is essentially trying to describe the transformation from the spatial temporal patterns of input spikes to the spatial temporal patterns of output spikes, as is shown in the bottom figure. Okay. So before we build the model, we ask ourselves the question, what do we really want from this model? Okay. And first, obviously, this model needs to be data-driven because this is a brain-machine interface. We want to interface directly with the brain signal. And second, it has to be predictive because we want to accurately predict the output signal generated by the hippocampus based on the input signal observed in the hippocampus. And in consideration of the characteristics of the hippocampal signal, such as the point process spikes, the spatial temporal nonlinear dynamics, and also the stochastic property. And also the model needs to be mathematically tractable, meaning that the model should be sufficiently constrained by the recorded data. And also this is something a lot of AI people won't agree, but I feel very strong about is that the model also needs to be biologically interpretable, meaning all the model variables should be related, right, to biological mechanisms or processes. So we end up with using this combined mechanistic and input-output modeling approach. Input-output modeling here means AI, right? To build accurate and interpretable model of the neural circuit. So we built two types of models. One is the MIMO model, describing the transformation from input region spikes to the output region spikes. It's essentially a point process time series regression model. And the second type of model, we try to relate the spatial temporal pattern spikes into memory content. For example, in this case, the specific category of an image the patient or the animals trying to remember. And that's essentially a point process classification model. Okay. So first, I talk about the MIMO model, the input-output model. This model structure is inspired by the biological principles you can observe in spiking neuron. It contains the feed-forward components describing the transformation from input spike to the synaptic potential, okay, like this. And it's described by a double convolution Volterra power series. And it can be related to biological mechanisms such as EPSP, IPSP, short-term synaptic plasticity, and dendritic integration. Okay. And then there's a pre-threshold noise and the threshold to generate stochastically the output spike. And this component is related to biological mechanisms such as neuronal noise, both intrinsically and extrinsically, and also the threshold determined by sodium and the potassium channel. Okay. And finally, there's a feed-forward kernel, right, describing the feedback dependent spike triggered after potential, right? As you can see here, right? So we design the model structure based on the biological principle, right? The stereotypical components of the spiking neuron. But for the more flexible components, we leave that to the AI model, so we can keep all the flexibility of those, input-output model components. So it's mathematically tractable, meaning all the parameters can be constrained just by the timing of the input spikes and output spikes. And at the same time, it's biologically interpretable because all the model variables can be directly or indirectly related to observable biological signals. That really facilitates the model interpretation and helps us to get better understanding to how signals are transmitted in those two brain regions. So we first test this model in animals. We trained animals to perform a memory-dependent task called delay non-matched sample task. The animal needs to remember specific locations and levels in order to get water reward. And at the bottom, you can see, based on our model, we can very accurately predict the output spike temporal pattern based on the input spike temporal pattern. On top, that's the actual recording. And at the bottom of each row, that's a predicted pattern. And also, we implement this model using VLSI with ASIC design. Okay. So here is the animal model we performed. In the beginning, on day one, we record normal C3, which is the input, and C1 output signal to build the model. And then we get the forgetting curve representing the memory function on right. And then we inject the drug to disrupt the input-output transmission, and the forgetting curve shifts down, right? And then finally, we use a model to bypass, to record from the input region, bypass the damaged region to generate the output signal. And by doing that, you can see we can at least partially restore the memory function, as you can see in the shifted memory forgetting curve. Okay. So we move forward to test this similar approach in human in collaboration with neurosurgeon and neurologists at Keck Hospital. And we use in human, we use a delay matched sample task. Basically, the patient needs to see images and try to remember those images. And we improve the model by adding a lot of the AI, right, machine learning components, and we get better prediction. And on left figure, you can see on top, that's the spatial temporal pattern recorded from the output region. And in the second row, that's what we, that's the actual one. And the second row, that's what we predicted based on the ongoing input signal and the computational model. And on right, we show that if we don't stimulate, right, or we stimulate the output region with random sequence, we won't influence the memory performance. But when we stimulate the output region with the model predicted spatial temporal patterns, we can significantly improve the memory performance, showing like the simulation does carry memory-related information. And by writing this information into the hippocampus, we can enhance the memory function. And we are also expanding this model by building deep MIMO model. This is inspired by convolutional neural network. Basically, we are treating the action potential as black and white images. And by doing that, adding the deep structure, we can capture arbitrary complex nonlinearity, and also we can share nonlinear dynamical components across different patients, across different tasks to enable transfer learning. Right. We also build that shallow memory decoding model because of the constraint of the data length. And we leverage machine learning methods like bagging and stacking, so we can include multi-temporal resolution features, and also we can assemble a lot of base learners into this meta learner to boost the model prediction performance. And eventually, what we show is based on the spatial temporal pattern recorded from the hippocampus, we can decode what kind of memory category the patient trying to remember. Basically, the category also the images, right? And so finally, I want to end my talk on this slide. So what I have been showing is the proof principle of the feasibility of building this kind of the model-based, right, hippocampal memory prosthesis. We test that in animal and human with a very specific behavioral task. So a lot of things need to be done in order to develop a real clinical viable memory prosthesis, including efforts in neural interface technology and the human test. But what I really want to emphasize here, there's still a lot need to be done in terms of computational modeling. And we can leverage a lot of existing AI technology for doing this. For example, this is already ongoing. We are building deep MIMO model, right? And in future, we will also build adaptive plastic MIMO model, right, to accommodate the change of the brain, right? And also we will build multi-task and multi-subject model, allow us to transfer memory or memory function across different patients. And that's really important for helping the patient with the brain damage. So I'll stop here.
Thank you very much, Don. The speakers have all made my job easy by giving very good talks and staying on time. So thank you for that. Now, if the audience could make the other part of my job easier and enter more questions so that when we get to the Q&A period, we'll be ready for that. Akhilesh is up next, and we're now moving from the brain to the retina, and in this case, artificial retina-inspired circuits.
Thank you, Steve. So we all heard some interesting talks about how electronics can help biology. Now we'll see the other way around, how biology can actually inspire electronics and create new kind of electronic circuits and help in computing. So I'll talk about this specific project that we have. Essentially, the idea is to create new retina-inspired cameras. So here is state-of-art machine vision pipeline. The basis of this is a CMOS camera. The CMOS camera generates frame-based data that is being processed by ML algorithms, usually deep learning algorithms. And this state-of-the-art pipeline creates multiple problems like excessive data transfer. You need excessive bandwidth because you are sending millions of pixels per frame for even a simplistic decision making, and it is also adversely susceptible. Now, it is not very, the existing pipeline, which is based on CMOS image sensor, has these problems. And one of the reasons is basically CMOS image sensors or cameras were never designed for machine intelligence. They were designed to really take good looking photographs. So we can definitely do better than that. So the intuition is to look at animal retina. And what we realize, at least from past decade of research in animal retina, is that animal retina has the recent research in retinal neuroscience has really changed the wisdom that retina is a passive filter that is doing some primitive filtering and sending data to the brain. In fact, retina is a very active feature extractor. It is expected retina is extracting almost around 40 different features in real time from the events that are happening in the surrounding and sending it to the brain. The brain is interpreting those features and giving us an illusion that we do have a camera in the eye. So essentially, the language of retina is feature spike. I can give a specific example. If an object is thrown at us, even before the brain can identify the object, retina has identified an approaching threat. and send a spike to the brain, indicating the brain that you should probably get out of this situation. So next slide. So the idea is really take retinal signs and put them inside existing manufacturable commercial camera technology. Now, retinal science in camera technology is not new. There is a good history to it. But interestingly, all of those technologies from knowledge from retinal science that has been translated into camera technology are based on late 80s understanding of retinal biology. The most recent understanding of retinal biology, some of these features, in fact, discovered probably a few months back, have not made their way inside electronic circuits and electronic commercial cameras. Anatomically, if I look at it, existing retinal signs in camera technology are dealing with the first photoreceptor and the first retinal synapse. Our proposal is really to take full computations from retinal synapse to retinal ganglion cells, the output of which becomes the input to the brain. And we are basically leveraging this recent thrust in the chip integration technology, essentially 3D integration, where we can integrate chips on top of each other and really map spatio-temporal computation similar to how computation is happening in different layers in the retina. So next slide. So let me take one specific example. This specific feature is called OMS, or object motion sensitivity. It is the ability of retina to identify moving objects in moving background. You can imagine how this could be useful for a camera that is sitting on a drone, because as the drone is flying, the entire receptive field is moving. And for surveillance purposes, you might be interested in objects that are moving in a moving receptive field. So shown on the left-hand side here is essentially the biological circuit in the retina that basically accomplishes this object motion sensitive feature. I'm not going to go in all the details there, but effectively what the retina is trying to do, it divides the entire receptive field into chunks of center and surround region. And then retina is constantly comparing the activity of cells in the surrounding region with that in the center region. If the activity of cells in the surrounding and center region are nearly same, retina assumes that there is no significant interesting object motion that is happening in the center region. If, however, there is relatively higher activity in the center region compared to the surround region, the retina assumes that this increased activity is created because there is a moving object with respect to the surrounding. So we took these understanding from through our retinal collaborators. These are experimental and theoretical retinal neuroscientists from Northwestern University. We first asked them to develop a software model. We then superimposed circuit constraints on their software model. And once we were convinced that these software models can really work on, large-scale complex videos extracted from real life, we then developed the circuits. Our circuits are based on APS pixel. These are the pixels that are currently in our smartphone, and also DBS pixel. This is the emerging area of dynamic vision sensors. So both with respect to RGB camera as well as to emerging field of DBS cameras, our circuits are amenable with both of them. Next slide, please. I can talk of another retinal feature here. The feature is called looming detection. Essentially, if an object is thrown at us, the retina can identify an object. In fact, there was an interesting experiment that was done wherein the looming detection circuit in a rat was deactivated, keeping the rest of the retina and the brain of the rat intact. And the rat significantly loses its ability to escape predators. So not only these, so there is a good sense that these features are actually controlling critical survival behavior in animals. Once again, we looked at the circuit, we created, with the help of our neuroscience collaborators, we created a software model, and then we implemented circuits that are amenable to mass manufacturing for commercial camera in standard semiconductor foundries. Next slide. So what we have here is two test frames from two specific videos from the dataset of BDD 100K, which is a self-driving dataset. The first video is runners running across a crosswalk and a car actually approaching the runners. So the receptive field is moving. So object motion sensitive feature in our iris camera can actually neglect everything else that's happening in the surrounding and can really differentiate that there are actually runners that are running through in this frame. The second video is basically a stationary car, and there is a car that is approaching this stationary car. So the loom detection circuit in our retina, it can neglect everything else and warn that there is an object in this region that is approaching you and is probably about to hit you. So you probably need to stop or take some action. So we now have these features that are implemented inside circuits that are amenable to the camera technology. Next slide. And the idea really here is, can we now think of next generation of computer vision where we can use these extra features that a camera can extract with deep learning, where you can have access to all the RGB features, all the RGB pixels, but along with that, you have rich context of additional features for the algorithm. We're also looking at new kind of spiking neural networks. All spiking neural networks today use two-dimensional feature, a feature in space and time. But now we are creating multi-dimensional feature because each feature has a specific meaning associated with it. So can we really create multi-dimensional spiking neural networks and also evolutionary algorithm? We are also creating a framework of co-evaluation where multiple agents can evolve together. And finally, I would end by just thanking all the collaborators. We have one-of-a-kind collaborative team consisting of hardware, algorithm, as well as neuroscience expert. This specific project was funded by Custom Exploratory Award, and we have a good set of advisors from Foundry as well as from DoD agencies and startups advising us on this project. And of course, thanking all the students who did all the heavy lifting here. Thank you.
Thank you, Akhilesh. So we're ready for questions now. My Zoom is struggling a little bit. I hope I'm coming through okay. I see the video is not, but I have it turned on.
Yeah, we can hear you.
Okay, good. We have 2 questions posted by Priyanka. So first of all, thank you, Priyanka, for the questions. You get the award for the most active questioner so far. And please keep them coming, and let's see if we can get questions from anybody else. I'm not sure exactly how the other sessions are doing this, but I guess I'll read the questions out loud in case anybody's not looking at the chat. The first one is for Maryam. It says from Priyanka, great application. Have there been proposed hypotheses or identified causes for variability on different responses?
Variability on different responses of, I assume they mean neural response either to input or to symptoms. So the hypothesis is that these variabilities, if you mean the variability in neural representations, could be state dependent. For example, if you think about a patient who is dealing with depression, depending on what their clinical depression diagnosis is, that state, that neuropsychiatric state may actually impact how their symptoms evolve over time, what the neural representations of the symptoms are. So there is some state dependence that dictates which networks are engaged and how they respond to stimulation.
Thank you. The next question is for Dong, again from Priyanka. The question is, amazing research, Dong. You mentioned different subject-specific future models. Could you share how you are thinking on this and potential advantages?
So what we have been using so far are all subject-specific models, meaning we build a model from data recorded from one animal or one subject and test that model within the same subject. So obviously, there's a lot of limitation of this approach, although this is useful for a proof of principle, right? For example, if we really want to help patients, and the patient very likely won't have normal input-output activity for us to build a model, right? So we have to use knowledge or data from other patients to build what I would call a subject like a general model, right? And one potential way of doing that is leveraging transfer learning techniques in AI, building this deep structure. So we can share intermediate features across different patients. But of course, in the end, we still need to have a fine-tuning, like subject-specific tuning for that model. But at least that gives the one possible solution for this problem.
Thank you. I have a third question from Priyanka, extending her lead over everybody else. This one is for Akhilesh. Great research, Akhilesh. What are additional applications your team has looked at, and what are constraints?
Okay, so our starting application was actually self-driving cars. That was because the availability of data set there. We are now looking into swarm of autonomous drones that can actually communicate with each other and agree upon some specific features that are critical for a specific task at hand. and those specific features would change as the task changes. So really looking at, can we go from application all the way to reconfiguring iris cameras and making it adaptive based on the specific task at need. The constraints that we really have in this particular case is actually coming from the circuit and hardware side, because the kind of spatio-temporal computing that retinal cells do are very rich and are difficult to mimic using normal transistor circuits. We got a bit lucky here because of the emergence of heterogeneous integration in chip technology. So it is helping us. Our workaround to that is basically to keep bugging our neuroscience collaborators and really ask them that what is the least amount of behavior you need from our circuit in order to extract this feature. We might not be replicating the entire biodynamics of these cells, but can we just do enough so that we are extracting these features? As a result, these features are not always very accurate, but they are degenerate. So the idea is, can the algorithm know that these features, we have degenerate set of features that can make mistakes once in a while, but can you really take those degenerate features and still get a robust decision making out of that? So there are multiple applications that we are looking at, specifically of interest to defense and DoD agencies.
Thank you, Akhilesh. I don't see another question submitted, so while I watch for those, I have a question of my own. I'm going to ask Yasser the next question. Yasser, if I understood correctly, you said that your printing techniques are about two orders of magnitude or two generations? I think you said two orders of magnitude.
So usually, if you think about state-of-the-art transistors, we talk about channel lengths in the range of 5 to 12 nanometers. With printing, you can actually go in the range of microns and even less. So, but like with even like the best electrohydrodynamic printing, you can go to hundreds of nanometer resolution. So essentially, you're off by 2 orders of magnitude, because channel length translate to your transistor performance and also how fast you can operate the devices.
Yep. And my confusion there, the answer was obvious. 2 generations, you'd be really small. But my question is, so that's how far you are today. And I assume there is room to scale and improve in printing. But I also assume the fundamental limits for printing are nowhere near the fundamental limits for lithography. I wonder if you can talk about that.
Absolutely. So it essentially, like the limitation comes from both the ink, because here you're dealing with liquids, and how fluid mechanics play the role on top of a substrate. Even if you drop the tiny volume of liquid, it's going to spread. And the best you could see is maybe hundreds of, not hundreds, 500 to 1 micron resolution with the electrohydrodynamic printing. And then there are tendencies to improve the surface. So the surface plays a role to even miniaturize the transistors.
So following up on that, yes, so you are limited by the liquid. So have you thought about the roll-to-roll printing for this purpose?
Yes, yes. So typically, how the process works, it's you start with some prototype printing with inkjet or blade coating, and then once your process is mature enough, then we go to roll-to-roll. Roll-to-roll takes up a good amount of ink and also materials, and the numbers I'm giving you in the microns, those are reported with roll-to-roll. And the benefit is you can think about meters per second speed in making devices. That's crazy. So it's like 1 meter full of transistors. Those are 1 micron gate length, channel length. And that gives you the scalability, but you will be limited by resolution.
Okay, good, thank you. Steve, can I have a quick question for Akhilesh? Yes.
Yep,
okay.
Please go ahead.
Yeah, so Akhilesh, I assume your hardware implementation so far is using CMOS technology. If that is true, have you thought about using emerging type of devices for the neural network, and what in your application, can you comment on that?
Yeah, so we have recently started looking into it, and definitely emerging devices are going to help our case. We first started with CMOS because we wanted to keep it at a level where it can be manufactured in industry today if the need be. But definitely, some of the constraints that we see from biology are difficult to achieve using CMOS devices. So emerging devices are a very good fit here. We were just constrained, by the maturity of these devices. And I know, Yoshua, you have been doing some excellent work there. So definitely something that we should follow up and sync up upon.
Thank you.
Okay, I think we've reached our scheduled break time. So unless anybody wants to insert one more quick question, actually we're at 6 after the hour. So I think it's break time. Yolanda, you want to jump in?
Thank you. Thank you, Steve. Yes, we will reconvene at, I believe,
11:15, right?
11:15, so 15 past the hour.
Mm-hmm.
Thank you, Joshua.
Great, yeah, thank you, Steve. Thank you for the speaker in the neuromorphic session. Let's have I guess 10 minutes break, and we will be back at 11:15. See you.